Hi everyone, welcome to our virtual classroom. Not going to go for high production value here, and we're not going to go all the way through chapter 8 today. What I want to do is just get you started and focus on some of the information that you'll need to get going on activity 1 on microbial genetics. The first part of the chapter, as we talked about, should be review for most of you. This should be the genetics you learned in Bio 101 or some other biology class. The learning outcomes for our first section are that you'll be able to define the terms genome and gene, to differentiate between genotype and phenotype, and to draw a segment of DNA and label all the important chemical groups within the molecule. You should also be able to summarize the steps of DNA replication and identify the enzymes used, and compare and contrast synthesis of the leading and lagging strands. So first, our genome. A genome is the sum total of the genetic material in an organism. Most genomes exist in the form of chromosomes, which are chromatin or DNA that's wrapped around proteins to organize it. We can also have plasmids. So as you see here in our bacterial cell, we have plasmids. In some fungus and protozoans, we can also get plasmids. What these are extra chromosomal pieces of DNA. So they're still packaged similar to chromosomes, but this chromosome, what we usually consider the organism's chromosome, either thread-like chromosomes in eukaryotes or a circular ring in prokaryotes. This contains all the information needed for the organism to function. These plasmids, our extra chromosomal piece, are just little segments, a gene or a few genes that contain information to build proteins that aren't necessary for the organism to function, but may offer some benefit. So our plasmids are extra chromosomal DNA. In eukaryotes and prokaryotes, our nucleic acid, our information for the genome, is contained as DNA, always. Viruses, not quite living organisms, as we talked about, but viruses can have either DNA or RNA, one or the other, not both. They can have double-stranded DNA, as shown here. Sometimes they only have a single strand of DNA. They can also have a single strand of RNA or a double strand of RNA. They're never going to have both DNA and RNA as their nucleic acid. Let's see, what else? So our chromosomes are DNA wound around proteins. Typically in eukaryotes, they are paired because we get our chromosomes half from our father, half from our mother. We call that diploid. Haploid would be a single chromosome, and some asexually reproducing protists have just unpaired chromosomes. Let's see. So the nature of our genetic material is that we have 3 different categories of genes. We consider our chromosomes, they carry our genes. Well, what exactly does that mean? A gene is a length along the chromosome that codes for a protein. We have 3 different categories of genes. Structural genes, these are the ones that actually code for a protein. This is what we think about, what we talked about, one gene, one protein. A gene codes for an enzyme that then builds some certain component or catalyzes a reaction for something that builds us, as we talked about. So structural genes code for proteins the body is going to use, like enzymes. We also have genes that code for the RNA, the ribonucleic acid, and that's the machinery that's used in protein production. So these don't actually result in a protein, but they have some role in producing proteins from our DNA instructions. And we'll get through that as we get to transcription and translation. And then we have regulatory genes. These are the ones that will control the expression of the gene. And by expression of the gene, we mean if a gene's expressed, we build that protein. So we can turn on or off the expression of genes. We'll talk more about that when we get to the lac operon. Some other terminology you should be familiar with. Genotype. That's the sum of all the gene types that we have. So we have a genotype made up of genetic information from mom and from dad for each one of our traits. That's our complete genotype. What information do our genes carry? And then the phenotype is the expression. What are the structures or functions we get from those genes building their proteins? All right, so DNA structure. If we look at our DNA structure, zoom in, this is back in chapter 1. The monomers of our DNA is a nucleic acid, that's our polymer. The monomers are nucleotides. So here is a nucleotide right here. It's made of a 5-carbon sugar. For DNA, it's deoxyribose. That's our 5-carbon sugar. We draw it like a pentagram because it's got 5 carbons. 1, 2, 3, 4. In this case, the 5th one is here off to the side. It's got an attached phosphate and then a nitrogen-containing base. So here is our nitrogen-containing base over here. On this strand, we see our sugar stacked in a row with the phosphate to the outside and our bases turned toward the inside. When we stack all of our nucleotides, so here's one nucleotide, here's another, we stack them all up side by side because this is a double-stranded structure. So we have one strand of monomer, 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 and a second strand, monomer, repeating monomers. So our polymer, repeating units of monomers, are 2 side-by-side stacks. If you think about Legos, you'd be putting 2, 1 here, 1 here, 1 here, 1 here, 2 side-by-side. To get a double-stranded with our monomers, and our monomers are stacked in a reverse direction. So here, this is a pretty horrible picture because it looks like our sugars are oriented the same way. But what this actually means, let me go back. What this actually means is in this second strand here, how I usually draw this is, this looks like a house, right? Our little pentagram here looks like a house. Here's the floor, here's the roof. One side, we pile our monomers floor to roof, floor to roof, floor to roof, floor to roof. This complementary strand would actually be flipped. So the roof would be on the bottom. My sugar would be oriented upside down. So here, where they drew the numbers, we see the 3' sugar is on the bottom. The 5, the peak of my house, let's say, is on the top. But on this side, the 3 is on the top, and the 5' sugar is on the bottom. Keep clicking on that. So really, I would draw that with that sugar, that 5-carbon pentagram sugar upside down to indicate that these are running in opposite directions. They are anti-parallel. So when we talk about the double helix, here's my sugar phosphate backbone on one side. Here's my sugar phosphate backbone on the other side. They run anti-parallel. My sugars are stacked upside down. So a really good analogy that a student in a previous class came up with for anti-parallel is that this would be like a 2-lane road. They're similar, but in one, the sugars face one way, and the other, the cars, or our sugars, go the other way, anti-parallel. So my sugar phosphate makes up the backbone, and we can think of this as being a ladder that's been twisted. So if I untwist this, I would have parallel backbones of sugar and phosphate, and the rungs of my ladder would be nitrogenous bases. Our bases, as we see, connect in the middle to give us one solid rung. And the way our bases pair up, we call this complementary base pairing, because only one base can pair with one other base. Adenine can only pair with thymine, and cytosine can only pair with guanine. Let's go back here. We see adenine and thymine, cytosine, guanine, thymine, adenine. I can only get this pairing because in order for these rails of my ladder, the uprights of the ladder, to be parallel, that means each of my rungs has to be the same distance. So we can see here, my cytosine is a single ring structure. My thymine is a single ring structure. My guanine is double ring, and my adenine is double ring. So the only way I can have parallel, the same distance across, is to have a single ring with a double ring. So I always get guanine with cytosine, even though I could have guanine with thymine. The reason I don't get that is if you see here, I've got 3 hydrogen bonds between guanine and cytosine, only 2 bonds, between thymine and adenine. I only have 2 places to form bonds here. So if I tried to bond my thymine with guanine, I would have this hydrogen out here looking for a place to bond, and it would result in a kink in my molecule. And so that wouldn't be as secure of a structure. So thymine only bonds with adenine, guanine with cytosine. So my complementary base pairing. The purines, these are my nitrogenous bases, are purines. Those are adenine and cytosine. I got those backwards. Sorry about that. The ones with a Y in them, thymine and cytosine, these are my pyrimidines. My purines are adenine and guanine, and I just remember which one by the Y. Sorry for that mistake in there. All right, so that's the structure of my DNA. Here's a better picture. Look, I made that same mistake again, sorry. So it's double helix, sugar phosphate backbone. Here's sugar, phosphate, sugar, phosphate, sugar, phosphate, running anti-parallel. This one goes 3' to 5'. This strand here goes 5' to 3'. And then I have cross-linked nucleotide bases, nitrogen-containing bases, with my purines and pyrimidines. Thymine and cytosine are my pyrimidines. Adenine and guanine are my purines. So let's get to activity 1. Part 1 asks for DNA replication. This question is the easiest question on this worksheet, and it's the most missed. So just think replication. Replication means making a copy of. I'm going to make a copy of my DNA. In the cell cycle, in eukaryotic cells, I have the S phase, the synthesis phase. I synthesize a copy of all my DNA so that when I go through mitosis and end up with 2 daughter cells, both of them have all the DNA. So I need to make a copy of it. So I have 2 molecules that I can then divide to 2 daughter cells, and both have all the DNA. So activity 1, part 1 is DNA replication activity. It gives you a double-stranded DNA molecule. These are anti-parallel. One runs 3' to 5', the other 5' to 3'. We have complementary base pairing. T and A always pair up. C and G always pair up. So whichever's on one strand, its complement is on the other. So this asks you to draw the results of replication. Well, if this is my original molecule, replication is going to result in 2 of these, and it asks you to label the original and the original strand and the parent. Here's my parent. And then which strand is new. If you go to question 2 and define semi-conservative replication, that should give you the answer for how to draw this and how to label it. So let's take a look at DNA replication. So overall replication, we talk about DNA replication as being semi-conservative. This is a very carefully orchestrated activity, has lots of different enzymes involved. We're just going to learn about 5 of these. The enzymes come in, they separate the strands, they uncoil the DNA, they separate the strands, they add new base pairs. So we end up with 2 daughter molecules. Each one is identical in their base pairing to the parent, but neither one of those new strands is completely new. Each of my 2, so I start with a double-stranded DNA. I separate them. I make copies using that complementary base pairing for my template. I now have 2 daughter molecules of DNA. Each one of these is identical to this original parent, but here these 2 are my original strands that I separated, and then I built new strands. So these are my new strands. So some of the enzymes that are involved in this process, topoisomerase is down here at the bottom, but that's the one that really comes in first. My DNA in eukaryotes is in chromosomes in the nucleus, and it's just sort of all spread out. If you think about like a skein of yarn just spread out all over, right? The skein is nice and wrapped up. Yeah, that's not how our DNA is usually. It's just all spread out. So topoisomerase kind of untangles that and helps to organize it a little bit. That's the first one that comes in. If you think about an old-fashioned telephone where the receiver is connected to the base, and it's got a helical connection, it's got a helical cord connecting the 2. If you used that phone for a long time and kept stretching out the cord and releasing it, stretching it, it all gets tangled up into a big mess. That's kind of what our DNA is like when it's in the nucleus, or in bacteria when it's in the nucleoid region. The topoisomerase would straighten that out, so now that cord would just be a single helical cord. The next enzyme that comes in is helicase, and that's going to unzip and unwind the cord. So it unwinds the helix, so I do have my ladder, and then it unzips by clipping those hydrogen bonds between the 2 nitrogenous bases. Primase, this is a primer. So primase is the enzyme that makes a primer, and the RNA primer, this is just a complementary piece of a molecule called RNA, that I'm going to talk about in a little bit, that comes in and latches onto the DNA, and its job is to tell our next enzyme, this is where you start making the new molecule. Here's where you start building. We have 2 different DNA polymerases involved in this. I'm not going to ask you to know the difference between 1 and 3. There are a bunch of DNA polymerases. So just DNA polymerase is the molecule. If we think about the name ACE, that ending ACE indicates we're talking about an enzyme. And in this case, we're talking about the enzyme ACE that builds the polymer DNA. DNA polymerase comes in and adds new nucleotides. So here's my original strand. I separate it out. DNA polymerase comes in, puts the complementary base pair nucleotide here, the next nucleotide next. So it runs around along the length of DNA building a new molecule by adding new nucleotides. When we're done with the process, DNA polymerase can also proofread. It's going to run up and down the new molecule to make sure everything is correct. And that prevents errors because our DNA is the information we need to function. We want it to be correct. The final enzyme that comes in is ligase. And what ligase does is stitches together any breaks in the DNA. And we'll talk about why there would be breaks. One reason why is if DNA polymerase found a mistake, it would clip out the mistake, the nucleotide that was put in incorrectly. It would put a new nucleotide in there, the correct one. But because it cut it out, it wouldn't be connected. Ligase would sew it into the molecule. We're also going to have segments on one of our strands. So let's look a little closer at replication. I've turned this image sideways so we can expand it a little bit. But if here's my original ladder, we see here it's uncoiled. Helicase has come in and uncoiled my molecule. Here, this is where helicase started, it was down here. It uncoiled and then it separated out by unzipping or clipping the hydrogen bonds between this nitrogenous base and its complement, between this and its complement. So helicase starts down here, runs up and untwists and breaks apart the molecule. Here's my original strand. Here's my other original strand. I've separated them out. I've put an RNA primer in down here off the picture to indicate, hey, DNA polymerase, this is where you start adding new nucleotides with complementary base pairings. So I know exactly what goes in here. If this is a T, then this must be an A. If this is a G, then this must be a C. And I can just run up and add those. And here's DNA polymerase just chugging along, adding bases. We call this side the leading strand. DNA polymerase can only move in one direction. So my primer, my RNA primer, is always put at the 3' end. That's my starting point, because DNA polymerase can only move 3' to 5'. So it's chugging along this way, right to left on this image. But my other strand is anti-parallel. So here's the 5' end. I can only put my primer down on a 3', and DNA polymerase can only move 3' to 5'. So it's building this from the 5' to 3'. It can't start here on this complementary strand and move this way because it can only move in one direction. Right? Anti-parallel, those cars going in opposite directions on the road. These cars go this way. These cars can only go this way. So what happens with this other strand, we call it the lagging strand because it takes a little bit longer to be synthesized, for the copy to be made, because we have to wait for helicase to open up my strand so that DNA polymerase can come in here. This would be 3'. So it could move this way from 3' to 5', but it has to wait till it has room to get in here. And then a primer will come in and say, hey, here, you can start here because this is available now. And the DNA polymerase will say, oh, okay, let me build here. Well, this section was available earlier, so it came in here and it built this. And then as more of the molecule opened up, it built that. And then this, and now as we open this, when there's room, another DNA polymerase will come and build this second segment. So what I get is on this lagging strand, I get short segments that have been synthesized, rather than here where synthesis is continuous. So it can happen faster. As soon as this opens up, my DNA polymerase can add more bases. So my lagging strand takes a little longer, and I have segments. So here I need my DNA ligase to come in and stitch those segments together. I'll post this abbreviated version of the PowerPoint that's already there, so you can click on these links and see an animation of DNA replication, see binary fission. I think I put these links into D2L already for you. The bacterial chromosome, because we're not moving in a thread, happens in two directions at the same time. So same enzymes, topoisomerase is going to unwind and relax the supercoils in my DNA. Helicase is going to cut the bonds between complementary base pairing. The replication fork is the point where I'm separating out my original DNA so that primase can come in give me a starting point, and then DNA polymerase adds new bases, but it's going to go in opposite directions until it meets up here, separates, and I have two daughter molecules. And then we'll have repairing and connecting with ligase. All right, so that's replication. I started with a molecule, I end up with two new molecules. So concept check, when DNA is replicated, two brand new nucleotide strands are produced. Is that true or false? What do you think? All right, that is false. We call this semi-conservative replication because when DNA is replicated, we have two new DNA molecules, but they are not both brand new. It's semi-conservative because for each daughter cell, here's my original or old strand. I separate it out, split my double helix, add new bases, and I end up with two identical strands. One was original. Nothing changed on this strand, but it was used as a template or a mold to tell me what new bases to add. Complementary base pairing. If this is A, the only thing I can put here is a T. If this is C, the only thing I can put here is a G, so I know what nucleotides to put in. And I make a new complementary strand. So I have one new strand, one old, semi-conservative replication. So now everybody should be ready to tackle activity 3, part 1. So we'll move on to section 8.2, where we are going to provide an overview of the relationship among DNA, RNA, and proteins, identify important structural and functional differences between RNA and DNA, draw a picture of the process of transcription, list 3 types of RNA directly involved in translation, define codon and anticodon, identify the location of the promoter, start codon, A and P sites during translation, and the E site too, and take a look at how these two processes differ in eukaryotes and prokaryotes. So this is going to address activity 1, part 2, and part 3. So there are a couple of questions here, 3 through 6 on from DNA to proteins. This has been changed on your activity form. So originally it said the coding strand in DNA shows 3 triplets. This is now the template strand. So my DNA has 2 parallel strands. One of them is the coding strand, one is the template strand. The template is what we build our RNA from. So I used the wrong one when I made this slide. The template, those first set of 3 triplets are the template. So I'm going to directly match complements. So it asks, what do I have to do first? So what's the term used to tell me what I do first to get from DNA to a protein? And then it asks what amino acids are coded for. Why are there 64 possible combinations of 3 nucleotides used, but only 20 amino acids? Why do we need that intermediate between DNA and a protein? And then define some terms. So we talked a little bit when we did our eukaryotic, how all those organelles work together, about this process of going from genes to proteins, from DNA to protein, where DNA is the complete recipe book. RNA gives us one single recipe out of that book. The protein is what we're making from that recipe. And the ribosome is the kitchen. That's where I actually bake my protein, bake the recipe. Gene expression is really a 2-step process that's involved in building a protein. When I say gene expression, that means to express a gene means to build what it calls for, which is the protein. So the central dogma of biology is 1 gene, 1 protein. Each of our genes codes for a protein. Usually that's an enzyme that's going to catalyze the chemical reactions that build the body. My 2 parts of gene expression are transcription, where I build a messenger RNA molecule from my DNA template, and translation, where I build the protein using the directions in that messenger RNA. So transcription, the master code of DNA, is used to synthesize an RNA molecule. And translation, that transcribed RNA, my messenger RNA, is used to produce a protein. Remember, a protein is a polypeptide made of a chain of amino acids. So my RNA codons tell me what amino acid goes in what order. For all eukaryotes, this is the order that we go in. DNA makes RNA, makes a protein. So similar enzymes, similar steps, we always start with DNA. We do have some exceptions because viruses can start with RNA. So they can sometimes take RNA and build other RNA. If it's an RNA virus, we're going to build RNA to be a template, to build more RNA, to be a template to build protein. We'll go through that in chapter 5. And we have retroviruses that actually come in with RNA and end up synthesizing a double-stranded DNA that will incorporate into the host cell. So much more on that. But we'll be coming back to all this in chapter 5. That's why it's important we get it here. We also have a lot of RNAs that are transcribed that don't actually code for a protein. Those were our regulatory RNAs. And those are used in the process sort of as machines that help. So they have different jobs in this whole process. So my genetic code, information on DNA, is based on a four-letter alphabet: adenine, thymine, cytosine, and guanine. I'm going to transcribe that into messenger RNA. RNA has some differences from DNA, and the most noticeable one here you'll see is that we don't have thymine anymore. We have a new nitrogen-containing base, uracil. Messenger RNA carries the code for how to build a protein that I can then translate using words made of 3 letters. So if we think of our 4 letters, our A, T, C, and G is the alphabet, or A, U, C, and G is our alphabet on RNA. All of my words are going to be 3-letter combinations of those 4 letters. And those 3-letter combinations, the triplets, are called codons. So quickly, we'll go step by step. The process of gene expression is I take the information in my double-stranded DNA. I use one strand of this as my template strand. So here is my template DNA, and I match up the corresponding complementary bases. G codes for C in RNA, C codes for G. A would normally code for T in DNA replication, but in transcription, when I build a different molecule, RNA, T still codes for A, so a T on my DNA gives me an A in my RNA, but an A on DNA gives me U, uracil, on my RNA. And then each 3 bases are a codon, one of my words, and those words can be translated into amino acids. So the genetic code is governed by 3 rules for codons. My triplets on messenger RNA are always read in one direction. So I can't read if this is my RNA, I can only read it in this direction. It doesn't reverse itself. The message is translated in a fixed reading frame, meaning 3 base pairs are read, and then I move to the next 3, and then in the next 3. I don't overlap, so it wouldn't be these 3, and then these 3, and then these 3, and then these 3. There's no overlap. 3, 3, 3. There are 64 possible 3-letter combinations. So I have 4 bases. How can I combine them into 3 different ways? That's 4 to the 3rd power, 64. We have 20 amino acids that are used to make proteins. So I only need 20 different codons, right? But I have 64. So what that tells us is that the genetic code is redundant. I actually have about 3 different ways on average that I can code for each amino acid. So when we say the code, those triplets are degenerate because there's wobble room in the third base. For most amino acids, or many of them, as long as I have the first 2 nucleotides, the first 2 nitrogen bases in the correct order, I can put anything in that third position and I'll still get the right amino acid. So that lets me have some variation, some mutation, without changing the message. Where that mutation comes from is DNA polymerase is really, really good about copying my DNA exactly. And if it makes a mistake, which happens about once every billion or 10 billion base pairs, I'll make a mistake. But DNA polymerase can also proofread, so it'll correct it. But every now and then, a mutation will get through, about every 10 to 100 billion base pair copies. So there's some variation, some mutation there. And then my RNA polymerase is the enzyme that builds RNA from DNA. It doesn't have any proofreading capability. So I can have mistakes in my RNA, even if there are none in my DNA. So that third base wobble room gives me some protection from errors in the transcription translation process. So here's my genetic code. This table's in your book. We'll look at it more closely. But if you count how many segments there are here are 64. And those are the unique ways I can combine my 4-letter alphabet into 3-letter words. And you'll also see, oh, look, I can get phenylalanine, it's abbreviated as PHE, 2 different ways. Leucine, 2 different ways. Oh, wait, look, here, leucine, 6 different ways. If I have these 2 bases correct, I can have some variation here and get the same thing. If I have these 2 first bases correct, I can have variation and get the right one. So there are 64. So on average, I have 3 different ways that I can get each one. That differs. Some are only 2, some are 4. I also have some instruction codons. AUG is the codon that codes for methionine, and it's also considered the start codon. Every polypeptide that will become a protein starts with methionine. Methionine isn't just the start codon. I can also have it elsewhere on my polypeptide, but we always start with methionine, with AUG as my start codon. There's a little marker on it that indicates this methionine is my start methionine, but if it's elsewhere in the polypeptide, it won't have that marker. And then when I get to the end of my polypeptide, when I have all my amino acids in a row in the correct order, I need to stop translation. And I have 3 different ways that I can stop translation. So a total of 4 instructional codons, which gives me 3 different ways, 60 amino acid codons, so 3 different ways I can get each one. Notice there is only 1 codon for methionine, so that I don't have mistakes in where I start or variation in where I start. So step 1 of gene expression is transcription. It's how we make RNA from a DNA template. The RNA that we make doesn't stay paired to the DNA. So in DNA replication, I started with my single molecule of DNA, double-stranded. I separate it and build 2 new DNA. The new strands stay connected. In DNA synthesis, I'll come in and I'll separate my strand out so that my RNA polymerase can come in and use this template strand to build the RNA. And then the DNA comes back together and the RNA goes out. In eukaryotes, it goes out of the nucleus to the ribosome to build a protein. And in prokaryotes, it just goes out to find a ribosome into the cytoplasm. So as I said, RNA polymerase has no proofreading, so this is not as accurate of a process as DNA synthesis. Here is transcription. There's my DNA. I'd have my replication bubble in a bacteria, and my RNA is produced from that and leaves the DNA, and then this will close back up. So here's my DNA. I have a coding strand. This is not the one that makes RNA. The template strand makes RNA. This is the template. My template always uses complementary base pairing. T codes for A codes for U in RNA, C codes for G codes for C. So complementary base pairing. When I build this, the template is the complement, because my RNA polymerase can only put an A, the complement, with a T. The reason this is called the coding strand is if I look at my base pairs on this, well, they're complements to the DNA. And these are complements to the DNA, except with U. So these are going to be similar strands, A. Oh, in RNA, my thymine is always substituted with uracil, G. So this has similar base pairing to this because it was complementary to my template strand. This is also complementary to my template strand. But my enzymes can't match an A with an A. So I couldn't use this to build this. So keep that terminology clear. Template strand, coding strand. Step 2 in the process, my nucleotides in my messenger RNA are translated into a sequence of amino acids, a polypeptide. Requires lots of energy, and I have 3 different types of RNA involved in this process. We just looked at messenger RNA. That's my message for how to build a protein, what amino acids go in what order. And I also have transfer and ribosomal RNA. These are the RNAs that we talked about that just have a mechanical or functional role. Transfer RNA carries my amino acids, brings them to the ribosome, and ribosomal RNA is actually a structural component of my ribosome. So if we look at, here's my messenger RNA, my transfer RNAs are going to carry amino acids. So this transfer RNA carries methionine. It doesn't know where to put that methionine, but it does know that it has the anticodon. That's not the codon. It doesn't tell me the amino acid. It's the opposite. It's the complement. So if we think of this like a lock and key, this anticodon is complementary to this codon. So the only place it can land on this length of messenger RNA is right here. So it lands here and it drops off methionine. This one finds its complement, lands there, drops off its leucine. So this has my message for the order of amino acids. This is what I look up on the codon table. Anticodon, not. Anti is not the codon. Don't look up this on the table. But the anticodon makes sure that the correct amino acid is put in the correct place. My tRNA, it is a length of RNA that was transcribed from a portion of DNA, but it folds back in on itself in these hairpin turns, leaving one segment down here that's complementary to its codon, the anticodon, so that it lands in the right spot. And up here has an acceptor stem. These codons allow the correct amino acid to be carried by this specific tRNA and only this tRNA. So the ribosome is composed, our ribosomes, cytoprotein synthesis, they're made of 3 molecules of ribosomal RNA. Again, it's RNA transcribed from DNA. And then they wrap that's wrapped around proteins. And it is going to chug along on our messenger RNA and add new amino acids. So here's my growing polypeptide. Keeps adding amino acids till I get this big long chain of amino acids. in bacteria, I can have lots of them running along my RNA at the same time. It's called a polyribosome or a polysome. So I can get a lot of protein, a lot of enzyme made really, really quickly when I need it. So the way I join those amino acids is with peptide bonds. It's called a polypeptide. That happens at the ribosome. The ribosome has 3 working sites, and think of it like an assembly line. The first working site is the A site, or the attachment site. That's where tRNA enters and brings in a new amino acid and starts the process. The second site that we go to is the P site. That P is for peptide bond, and that's if I have an amino acid come into the A site, and then I move that to the P site, a new amino acid, or tRNA, comes in, and then those 2 are joined with a peptide bond. And then the E site is the third site. The tRNA is released. That's the exit site. And we just keep moving along, adding new amino acids as we go until we get a big long chain. So here is my actual ribosome. This gray in the background and this blue in the background are RNA molecules. And then these other lavender colors are the proteins that are wrapped around to give it the exact right shape to do this. The subunit here, the smaller subunit, this is where the messenger RNA latches on and gets fed. Well, the ribosome feeds along the messenger RNA. And then the top site has the A, the attachment. My tRNA comes in, latches on, attaches, and then it shifts to the P site where I can form a peptide bond between these 2 amino acids. And then that tRNA comes here, the exit site, and it leaves, but my amino acids are connected. So I get a growing polypeptide. So activity 2, again, activity 1, part 2, that second DNA to proteins. You should be able to answer these questions now. Activity 3, as you continue on, transcription, translation, you should be able to do this. I just want to show this one activity. This is really a fun puzzle. It's an exercise in complementary base pairing. So we know DNA complements. A always goes to T always goes to A goes to T. Well, whichever side I know the complement. G goes to C goes to G. So I know this triplet is CGG because it's the complement of this one. I know this triplet is TAT because it's the complement of this. And I can do the same here and here. How do I get these though? Oh, well, messenger RNA is made through complementary base pairing from my template strand. And here we say this is the strand that will be transcribed. So this is my template strand. So I know GCC will code for CGG. I know ACT, well, A codes for uracil in messenger RNA. So here will be U, C will be G, and T still codes for A. So I can figure out my messenger RNA from the codons I have here, but I can figure out these blank spaces because these are the complements. My U, my uracil in RNA is coded for by A in DNA. My A is coded for by T. My U coded for by A. So ATA. U, well, that's going to be coded for by A. I have AA here, then I know its complement in DNA is TTT. I still have one spot that I'm not sure of, but I have the anticodon, which is the complement in RNA. U is coded for by A. A is coded for by U because I'm matching RNAs, and C is complementary to G. Now that I have my messenger RNA codon, I can figure out what my complement in DNA is, and then its complement in DNA. And then I'm going to use my codon table to identify the amino acids. And there's a warning here. Remember which molecule carries the code as a series of codons. This is my message with codons. This has anticodons, not the codons. This is just kind of a fun puzzle for now because in eukaryotes and prokaryotes, we always start with DNA, double-stranded molecule. A template strand gives us our messenger RNA. Our tRNA doesn't have any message. These are just individual molecules that come in and match up with their complementary base pairs on messenger RNA. But I can match them. So just an exercise in complementary base pairing. My amino acids come from the codons on messenger RNA. So I'll go look for those codons here and put those into the right order. That should get you started and through our first part of the worksheet. That's the first part of the chapter, so going through replication and gene expression. Some differences between transcription and translation. In bacteria, the start codon's always AUG. In eukaryotes, we can have different forms of methionine for the start methionine and for others. Messenger RNA can code for several genes, one gene, one protein for us. We can have both of those going on simultaneously in the cytoplasm of bacteria, transcription and translation. Because our DNA is in the nucleus, transcription can only occur in the nucleus. Because we need DNA and our DNA never leaves the nucleus, translation can only occur out in the cytoplasm at a ribosome, either on the endoplasmic reticulum or floating around in the cytoplasm, but our ribosomes are out in the cytoplasm, outside of the nucleus. And then our genes exist as an uninterrupted set of triplets coding for protein. Eukaryotes a little more complex. All right, final concept check. Which of the following types of RNA contains hairpin loops and is involved in carrying amino acids to the ribosome? Messenger RNA, regulatory RNA, ribosomal RNA, transfer RNA, or primer RNA? Right, carries, that's our transfer RNA. All right, I hope that helped. Watch the videos. They are better than me being a talking head with PowerPoint. They'll help you get through that. And I'll post a second video for the second part of the chapter soon. How do I stop recording? Let's go find Loom.